Thank you very much for kind introduction, also for inviting me here. Thanks to the organizers. And as you already introduced myself, I'm a physicist by training, but we move more and more into biology. So the group is doing biophysics and material science, and so the talk will be probably different than the others you have heard today. But the aim is that at the end I can maybe introduce two techniques which might be also helpful for cancer research at the end. Well, the MPI is not anymore called metals research, it's called intelligent systems, and so the name changed. And it was somehow easy because nothing can be said against intelligence. But the group is also, nearly half of the group is located here in Heidelberg since more than 10 years in the Institute for Physical Chemistry. So the group explores interactions of cells with biointerfaces, where we can tailor them in a very specific way. And we also develop techniques like optical techniques to identify certain phenotypes, as I will talk today about it. And if you look at cells at interfaces, your first obvious thing is, of course, adhesion. So if you look at tissue cells, these are standard fibroblast cells adhering to interfaces. And we marked here vinculin, a protein of the focal adhesion cluster. And by looking at these types of adhesion processes, here still marked with a fluorescent molecule so that you can identify the adhesion sites, you can ask the question if maybe you can track out of these images, out of these adhesion processes, some very specific for a certain phenotype, and maybe even do this at the end marker-free. So it should be quick and it should be marker-free. And if we then leave fluorescence microscopy and we go to a actually very, I would say, rather primitive optical microscopy technique, which is known since more than 20 years, this is called reflections interference contrast microscopy. And what this microscopy actually does is it especially highlights adhesion areas by looking at the interference of two light waves. One is coming in, this is this one, and the other one is the reflective light, and these two waves, they interfere with each other, and what you eventually can do very easily is then measure distances. You can measure the distance of a cell membrane from the substrate, and if you can do so, you can also identify very easily where the membrane is bound to the substrate through adhesion and where it's not the case. This is working without markers. It's just using a standard microscope, which probably every one of you have in your lab. You see here the comparison. First of all, this is a cell which is adhering to a substrate recorded by this reflections interference microscopy technique. And you see here dark areas, you see more bright areas, and the measure for the darkness or the brightness is a measure for the height. So from this different level of brightness, we can directly say measure the height of the membrane from the substrate, and this goes with a resolution in the nanometer range. And this also allows you obviously then a very strong contrast for very small structures like lamellipodia formation, as you see them here. While if you compare them with bright field imaging, this is somehow very, very difficult and of course not as precisely detectable. But if you have now such a high contrast with respect to adhesion, then you can develop certain imaging analysis tools to identify these cell structures very easily with a software, and you can then track out the cell area. You can track out the cell adhesion area, or the full cell area here, the adhesion area, the black spots, and you can also very precisely measure the contour of a cell. And this is what I will come to, is now to concentrate. If you take now, let's say this is two model cell lines from pancreas cancer, PAT2T and PAT2S, where Pat2T is more aggressive than Pat2S, then you can see that just by looking at the contour of such cells, you can see a distinct difference. So you can really pin down a phenotype. And these differences in contour, well, this one, which is more invasive, looks more complex, there are more tiny structures, so it's not as well defined as in the bottom case. And this kind of irregularity can be measured by a mathematical model, so to say, which is called fractal geometry or fractal dimensions. And fractal structures is something very, which can be observed very often in nature, and there are some, if you look at plants, but also if you look at brain or if you look at tissue. So if you want to describe all these geometries, fractal dimension is a way to do so. And the higher, the larger the complexity is, the higher at the end the fractal dimension is. And I avoid now to go into the mathematics of this, but the message is, so the more complex a structure is, the higher is the fractal dimension, and it's at the end a measure for irregularity. Now if you look into cancer, these are this is melanoma, a normal and a cancerous. And if you just look on the macroscopic scale, you already see that the irregularity is obviously much higher in the cancerous type. That's how you identify skin cancer. But do we actually also see this on the single cell level? So if you extract cells from tissue and you characterize the geometry, can you at the end get a measure of the aggressiveness of the tumor? And so we use these for the beginning, these model cell lines, which are different in their invasiveness. So Pat2T is more aggressive than Pat2S. And we wanted to see if we can quantify the difference in aggressiveness by measuring the fractal dimensions with this rather easy optical microscopy, the reflections interference contrast microscopy. And these are now two examples where we measured the contour. This is Pat2S and this is a Pat2T cell. We measured also the variation of membrane height, which we see here in the different gray scales. And you can also combine these two measures, and these are two independent measures to get the combination out of this. And if we measure this, then you see here the outcome. We see here the number of cells as a function of the fractal dimension. And in the case of Pat2S, which is the less aggressive one, you see here a fractal dimension which is lower than in the case of Pat2T. And so there's a significantly higher fractal dimensions for the metastatic tumor cell line in this particular cell type. What we then did is we compared this with, so we tested somehow what is the probability that you can actually measure with fractal dimensions that a Pat2T cell is a Pat2T and not a Pat2S and vice versa. And what we did is we labeled, we took Pat2T and Pat2S, we labeled Pat2S with red color and Pat2T with blue color, so we actually knew what it was. Then we mixed them together, and then you just used reflections interference contrast microscopy and the measuring of the fractal dimensions to identify Pat2T and Pat2S just by fractal dimension. And if you just take the contour that's shown here, then you already see there's a distinct success rate. This is the measure that you get Pat2S correctly. So it's a true positive, and there's about 30% true negative. But if you then take all the the Pat2T cells, the aggressive ones, and then if you revisit your sample and look at all the Pat2S cells and measure it again with the topology, so the second independent measure, then you can lower the chance of not identifying a Pat2T cell to be a Pat2T cell down to 3%. And if you compare this, if you use standard antibody labeling techniques, in literature you find this value to be around 15%. So, of course, these values can be discussed, and they might be quite similar, But what this technique, measuring fractal dimensions with this easy microscopy technique offers is that it's very easy, very quick, and it can, in the end, very quickly installed in any laboratory. We then went further to other pancreas tumor cell lines. We looked here at, for instance, CAPAN2, CAPAN1, PA2S, PA2T, and PANC1, which varies in their aggressiveness. PA2T is known to be the most aggressive one, and CAPAN2, for instance, is the least aggressive one, and then there are intermediate ones. And if you then scale the fractal dimension, especially the contour, then you see in this prognostic map, how we call it, we plotted here the fractal dimension of topology against contour. You can then really pin down for each of these specific cell lines a parameter which identifies the cell line, and you can say the higher the contour, the fractal dimension contour of the contour is, the higher is the aggressiveness. You have your PA2T, which is the most aggressive one, then PANC1, CAPAN1, PA2S, and then CAPAN2. So this is fine. This was all so far just with cell lines, and of course we are now going into primary cells. We also collaborate with clinicians to get tumor samples, which we have to do in collaboration, of course. And then we try to correlate the fractal dimensions with invasiveness of these tumor cells. And the other thing, what can also add in addition to use now for the measurements we have done so far, we just use let's say fibronectin-coated substrate, or just a glass plate uncoated. But you could make this much more specific by developing biointerfaces which then presents in a very well-defined ligand for, let's say, E-cadherins or for a certain integrin, which is expected to be highly expressed in a certain expected tumor cell. Just to give you an example that these surfaces can actually really trigger a certain phenotype, I just show you here two tissue cells where the only difference is it's the same tissue cell. The difference is that you have here a very specific ligand for integrin alpha V beta 3. And then you can change the spacing very, very slightly, so you change the concentration and you change the phenotype completely. So if you then would build a substrate which offers specific ligands or antibodies for a certain tumor cell you expect, then you can actually even enhance the contrast which you expect from measuring the fractal dimension. And by just changing the substrate, you see how you change the phenotype. This is just fluctuations of the adhesion contacts. Just if you change the density, you can make them very stable or you can make them very dynamic. Also, this is a very specific marker for identifying cell types. Another marker, which is also some marker-free ways at the end to measure forces. So it's not just measuring adhesion contacts and the structure of the contour. In addition, you could easily measure the force which a cell brings to a substrate, and it will be different and very specific to the different tumor types we are looking at. Finally, I come to the last point, how we measure invasiveness of cells. And this is a project which goes back 10 years from today, which we have done together with Thomas Seuferlein from the Internal Medicine in Ulm on pancreas cancer cells. And he motivated this work by the fact that he says in the pancreas cells, in the case of primary tumors, you find less SPC, while if already pancreas cancer has formed metastasis, you have a higher amount of this SPC which can be measured with patients. And then we got interested in why this SPC seems to be a marker for aggressiveness. Then we started just to look at the intermediate filaments of these panc cells, where you see that by adding SPC, you see a change in the structure of the intermediate filaments. So this is the nucleus, the membrane is out there, and you see here the intermediate filaments, and somehow changing to a perinuclear, it's somehow wrapped, the cytoskeleton wrapped around the nucleus. And if you draw this schematically, what you see is a change from a structure which fills the cytosol to a structure where the cytosol is somehow empty from intermediate filaments. And you can then ask, is maybe the structure, is the cell softer? So is stiffness a measure, stiffness of a cell a measure for aggressiveness? Then how to measure stiffness of a single cell? We built then so you cannot do it with hands like you would do it maybe with a piece of rubber. But you can do it with smaller hands, with small microplates. They are in the micro dimensions, and this allows us to stretch a single cell and to measure the force you need with this. And if you have these two values, you can give a elastic constant, which we monitored here, and then we measure the elasticity over time. And then if you add SPC, then you see a drop in the elasticity, so the cell becomes much softer. And this is associated with the rearrangement of these intermediate filaments. So stiffness is a measure, in this case, for aggressiveness. The question was still, is this also true that these cells can penetrate tissue quicker? And also here we went into a model system, an in vitro model. It's a microfluidic-based system where we built a chamber where the left side is filled with the cells of interest, and they need to migrate through small channels, which are plated here, to the other side. And the channels, they look like you see them here. So this is the left side, this is the right side, and the cells migrate through. And now we can vary the width of these channels. We can make them wide or small, and we can make them so small that actually cells do not migrate through. And this is the example here. These are the Planck cells in the case where there is no SPC added to the media. And you see here the cells, they penetrate, and we track here by the color points the lamellipodia where the cell tries to invade these channels, but they somehow do not manage, so no cells on the other side. But in the same situation, now we add this SPC, which causes this change in the intermediate filaments structure, which causes the change in elasticity. So by adding 10 micromolar of SPC, you actually find then cells really to manage to migrate to the other side. And you see this with high optical resolution. You can even then look onto these behaviors with fluorescence. As is shown here, it's the nucleus, it's the actin, it's the intermediate filaments. Why this is so organized in a structured way in the channel, we don't know. That's we are looking into. But more importantly, if you look at the statistics, so if you look at these penetrations of these channels, and there are hundreds of these channels which you can read out automatically, so you get strong statistics. You see here on the left side is the situation at the Planck cells without SPC, and you see only a small portion of 10% which really make it to the other side, which is the permeation. But if you add SPC, then this goes up, so more cells penetrate to the other side. So that's how we quantify somehow in the lab invasiveness. Of course, there are other ones and more in vivo relevant, but this somehow allows really to look also on molecular mechanisms, what's happening in such tiny pores with certain molecules or structures like the intermediate filaments. With this I end. I want to thank you for your attention, of course. A lot of the work has been done in collaborations like with Benjamin Geiger from the Weizmann Institute or Horst Kessler from Munich and others, and this is the group who, of course, did the work. Thank you very much.
Thank you very much. Are there questions? Yeah.
Can you do fractal dimension analysis on fixed cells in tissue? Does it have to be free cells?
No, it can be fixed. It can also be tissue. It doesn't need to be on a substrate. Yeah. So in general, I think it's worthwhile to look also really in tissue onto the fractal dimension of cells to get a objective measure of the phenotype at the end. Yeah.
And do you know if you take a primary tumor, do it in the tissue, and then dissociate the tissue and look at the cells individually, it's the sort of relative order of fractal dimension for cells in different tissues?
We have not done that, but something we would like to do which is not easy for us because we have no patients, but we do this in collaboration in the future.
So if there are no further questions, I would like to thank all the speakers for their exciting presentations.